One final evidence that oxygen was not ordinarily present in the early atmosphere and, therefore, was not dissolved in early oceans, is that organic compounds are stable in the presence of hydrogen, that is, under reducing conditions but are quickly destroyed in the presence of oxygen. Haldane, 1933. This indicates that compounds required as precursors or building blocks for living systems must have originated and accumulated in a reducing environment. Primitive C served as broth for the prebiotic synthesis. Classical experiment of Miller, 1953, has established that organic compounds can be formed without the intervention of living organisms. Thus, it appears likely that the sea of the primitive Earth spontaneously accumulated a rich mixture of organic molecules. In the absence of living organisms and oxygen, the organic compounds would have been stable and would persist for countless years. The sea became a sort of dilute organic soup or broth, or aquatic garden of Eden of Volpe, 1985, or chemical cauldron of Watson et al., 1986. In the organic soup of cauldron, the molecules collided and reacted to form new molecules of increasing levels of complexity. Proteins capable of catalysis or enzymatic activity had to evolve, and nucleic acid molecules capable of self-replication must also have developed. 4. Evolution of progenote. The procellular evolution involved the following three main steps. I. Origin and evolution of RNA world. The living cell is an orderly system of chemical reactions, which are directed by enzymes, that has the ability of reproduction. Current consensus is that the machinery for self-replication, that is self-duplicating nucleic acids, evolved before the development of the metabolic, enzymatic, machinery of the cell. It is likely that self-replicating polynucleotides, nucleic acids, slowly became established in the primordial Earth some 3.5 billion years ago. In the absence of proteinaceous enzymes in the prebiotic environment, less efficient catalysts in the form of minerals or metal ions would be sufficient to promote the reactivity of the nucleotide precursors. It is postulated that errors continually occurred in the copying process and that sequence of nucleotides in the original polynucleotide molecule became altered on many occasions. Large numbers of polynucleotide variations undoubtedly maladapted. Just as organisms today compete for available resources, molecules with different nucleotide sequences competed for the available nucleotide precursors in promoting copies of themselves. Any new mutant sequence with a replication rate higher than the antecedent sequence could be fittest and prevail. Natural selection, or differential reproduction, thus, operated on population of molecules as it did later on population of organism. The highlights of RNA would include I. Distinction between genomic and functional RNA molecules, E. Recruitment of exons into transcription, E. Inclusion of introns in most genes, IV. No regular cell growth, and B. Formation of membranes, figure 12.5. E. Origin and evolution of ribonucleoprotein, RNP, world. Certain polynucleotides not only specified their own sequences, but apparently directed the synthesis of macromolecules of an entirely different type, namely, polypeptides, proteins. In present-day organisms, RNA guides protein synthesis, which suggests that the nucleotides of RNA, not DNA, were the first carriers of genetic information. Thus, RNA established a primitive genetic code for ordering amino acids into proteins. Evidently, the genetic code, the translation of nucleotide sequences into amino acid sequences, became established at a very early stage of organic evolution. The universality of the genetic code in present-day organisms attests to the early origin of genetic code. The highlights of RNP world include I. Origin of primitive tRNA, rRNA and aminoacyl tRNA synthetase enzyme, E. Evolution of template-dependent translation apparatus, true mRNA, and transcription and replication of segmented double-stranded RNA genomes. E. Origin of plasma membrane. A selective advantageous RNA molecule would be one that directs the synthesis of a protein that accelerates the replication of that particular RNA, i.e., RNA polymerase. However, a protein specified by a particular variant of RNA could not foster the reproduction of that kind of RNA unless it is retained in the immediate vicinity of the RNA. The Free 154 EVOLUTION, Evolutionary Biology. Diffusion of proteins could be prevented if some form of a compartment evolved to enclose or circumscribe the specific protein made by a particular RNA. All present-day cells have a limiting plasma membrane composed principally of phospholipids and proteins. Most probably the first cell was formed when polarized films of phospholipids formed soap-like bubbles enclosing aggregations of complex macromolecules. Specifically, a membranous structure arose that encircled a self-replicating aggregation of RNA and protein molecules. Once bounded by alamiting membrane, a given RNA molecule could be assured of propagating its own special protein molecules. IV. DNA world. At some later stage in the evolutionary process, DNA took the place of RNA as theropository of the genetis information. This step may have occurred due to evolution of an enzyme called inverse transcriptase. In other words, the genetic code was stored in DNA rather than RNA. Today, RNA molecules serve essentially their primal function, directing protein synthesis. Thus, in modern cells, genetic information is stored in DNA, transcribed into RNA, and translated into protein. An endless permutation of nucleic acids and proteins has fostered an enormous richness of life. Thus, evolutionary highlights of DNA world include I. Evolution of multiple pieces of DNA, E. Stable storage of information in DNA, E. Sure existence of plasma membrane, and IV. Growth and division. V. Origin of progenote. The procellular stage of organization has been termed progenote. The nature of progenote is uncertain. It might have been a functional, slow-replicating, independently evolving organism, or it might have been an amorphous group of primitive transcription units plus A** primitive transcription and translation apparatus from which many different cell lineages evolved, 3 of which have survived to the present day, i.e., archaebacteria, eukaryote and eubacteria. The progenote contained DNA, archaebacteria. Most genes with introns, a slow growth and heterophic nutrition. The first living system, the progenote, drew upon the wealth of organic materials in the sea broth. Organisms that are nutritionally dependent on their environment for ready-made organic substances are called heterotrophs. The primitive one-celled heterotroph probably had little more than a few genes, a few proteins and a limiting plasma membrane. The heterotrophs multiplied rapidly in an environment with a copious supply of bacteria. Dissolved organic substances. However, the ancient heterotrophs could survive only as long as the existing store of organic molecules lasted. Eventually, living systems evolved the ability to synthesize their own organic requirements from simple inorganic substances. Thus, in the course of time, autotrophs arose, which were able to manufacture organic nutrients from simpler molecules. However, primitive autotrophs would require a whole array of enzymes to direct a multi-step chain of metabolic reactions involved in the synthesis of a protein. Indeed, it would be too much to expect that all the necessary enzymes evolved at the same time. Norman H. Horowitz, 1959, has proposed that the chain of steps in a complicated chemical pathway evolved backward. Let us analyze his concept in the following way. Retrograde evolution. According to Horowitz, an organism might acquire, by successive mutations, the enzymes required in a biosynthetic pathway in the reverse order from that in which they normally appear in a sequence. In other words, evolution began with the end product of the pathway and worked backward, one step at a time, toward the beginning of the reaction chain. The total pathway became constructed when the organism had tapped from its surroundings all of the intermediate precursors of the reaction. For instance, let us suppose that an organic compound O is synthesized through the following steps, where A through D represent precursors. A greater than B greater than C greater than D greater than O. Origin of life 155. The first primitive heterotroph, lacking synthetic ability, would require the presence of O in its environment. This essential organic compound, continually being used by the heterotroph, would ultimately become rare, if not exhausted. If a mutation occurred that granted the heterotroph an ability to catalyze the reaction from D to O, then the organism would no longer remain dependent on the availability of O in the environment. Indeed, in an environment where O had become depleted, the new mutant would have a survival or selective advantage over the ancestral type and replace it. As the new mutant reproduced, D in turn would become scarce. Another mutation might occur in the mutant, converting it to a still newer form capable of catalyzing the reaction C to D at this juncture. Both the first mutant and the newly arisen mutant could live together in a close mutual, symbiotic relationship, establishing the first two gene systems. The intimate two-gene combination would be able to survive and reproduce in the presence of C and D as other compounds in synthetic pathway become progressively rarer. Additional mutations for their synthesis would be favorably selected. Ultimately, a multigenic system would evolve capable of directing the synthesis of O from inorganic substance A by way of intermediate products B, C, D. At first glance, it might appear that an unreasonable number of mutational events has been involved. But it should be recalled that mutations occur normally and continually in living organism. In fact, the capacity of mutation should be regarded as an essential property of life. V. Adaptive radiation of progenote. The first simple autotroph arose in an anaerobic world, one in which little free oxygen was available. The primitive autotrophs obtained their energy from the relatively inefficient process of fermentation, the breakdown of organic compounds in the absence of oxygen. In addition to provide available energy in the form of ATP, such fermentative reactions could also be developed to provide reducing power in the form of NADH that could be used to derive biosynthetic reaction, figure 12.6. Glucose plus ADP greater than lactate plus ATP, e.g., lactobacillus glucose plus ADP greater than ethanol plus lactate plus co, plus ATP, e.g., leuconostoc glucose plus ADP ethanol plus co, plus ATP, e.g., zymomonas IV, lactate plus ADP greater than propionate plus acetate plus ATP plus co, e.g., Clostridium. Figure 12.6. Some fermentation reactions producing ATP and examples of prokaryotes which perform them, after King, 1986. Thus, the early fermentative autotrophs were much like our present-day anaerobic bacteria, lactobacillus, etc., and yeast. The metabolic processes of the anaerobic autotrophs resulted in the liberation of large amount of carbon dioxide, CO, into the atmosphere. Once this occurred, the way was paved for the evolution of organisms that could use Colorado as the sole source of carbon in synthesizing organic compounds and could use sunlight as the only source of energy. Such organisms would be the photosynthetic cells. Early photosynthetic cells probably split hydrogen-containing compounds, such as hydrogen sulfide H, S. In other words, as is electron micrograph of lactobacillus. Still performed today by anaerobic green purple sulfur bacteria. Hydrogen sulfide is cleaved into hydrogen and sulfur. The hydrogen is used 156 evolution evolutionary biology by the cell to synthesize organic compounds, and the sulfur is released as a waste product, as evidenced by the Earth's great sulfur deposits. In time, the process of photosynthesis was refined so that water H0O served as the source of hydrogen. The result was the release of oxygen O as a waste product. At this stage, free oxygen became established for the first time in the atmosphere. The first organisms to use water as the source of hydrogen in photosynthesis were the blue-green algae, cyanobacteria. Since blue-green algae were active photosynthesizers, atmospheric oxygen accumulated in increasing amounts, converting reducing primitive atmosphere into oxidizing atmosphere. Oxygen would be toxic to anaerobic bacteria. Many primitive anaerobic bacteria, incapable of adapting to free oxygen, remained in portions of environment that were anaerobic, such as sulfur springs and oxygen-free muds. Today, there are bacterial types that are anaerobic as well as aerobic. Geologic evidence indicates that free oxygen began to accumulate in the atmosphere about 2 billion years ago. Terracing levels of atmospheric oxygen set the stage for the appearance of one-celled eukaryotic organisms, which arose at least 1 billion years ago. Then, within the comparatively short span of last 600 million years, the one-celled eukaryotes evolved in various directions to give rise wealth of multicellular life forms inhabiting the Earth. Evolution of eukaryotes. The simplest cells in nature, the bacteria and blue-green algae, cyanobacteria, have been classified as prokaryotic cells. These cells have no nuclear membrane by which the hereditary materials, DNA, are set apart from the cytoplasm, and lack specialized cytoplasmic bodies, organelles, such as mitochondria and chloroplasts. In a bacterial cell, the DNA molecule, which does not contain introns but does have operons, forms a simple closed loop that is attached to the inside of the plasma membrane. The replication of a bacterial cell occurs rapidly by the simple division of the cell into two. To recall, phylogenetically, the progenote evolved into 3 types of cells: 1. Archaebacteria, 2. Eubacteria, and 3. Eukaryote. Archaebacteria are the prokaryotes which appear to have diverged from the true bacteria, or eubacteria, berry. Early in evolution, figure 12.7. The strange class of Archaebacteria consists of bacteria that are tolerant of acid, thermophiles or thermoacidophiles. A cyanobacterium, Anabaena, bacteria that thrive in salt, halophils, and bacteria that generate methane, methanogens. Recent developments in molecular biology reveal that these bacteria share some striking biochemical features, such as 16S rRNA sequence, that are absent from other bacteria. Eubacteria includes cyanobacteria aerobic, H. O photosynthetic chloroxybacteria aerobic, H. O photosynthetic Paracoccus aerobic, heterotrophic non-sulfur bacteria aerobic, H. S photosynthetic sulfur bacteria anaerobic, H. S photosynthetic green filamentous bacteria aerobic, H. S photosynthetic green sulfur bacteria anaerobic, H. S photosynthetic spirochetes anaerobic. Eubacteria includes cyanobacteria aerobic, H. O photosynthetic chloroxybacteria aerobic, H. O photosynthetic Paracoccus aerobic, heterotrophic non-sulfur bacteria aerobic, H. S photosynthetic sulfur bacteria anaerobic, H. S photosynthetic green filamentous bacteria aerobic, H. S photosynthetic green sulfur bacteria anaerobic, H. S photosynthetic spirochetes anaerobic, heterotrophic Clostridia anaerobic, heterotrophic, and disulfovibrio, sulfate respiration, C. Slay. 1986. The eukaryote is the ancestor of modern eukaryote. In contrast to the prokaryotes, the cells of the morphologically more complex plants and animals, or eukaryotic cells, have a distinct nuclear membrane that encloses strands of DNA, or discrete chromosomes. Eukaryotic cells also have an elaborate system of membrane-bound cytoplasmic organelles. The enzymes that are responsible for releasing energy from complex organic molecules are packed inside sausage-shaped organelles called mitochondria. In the eukaryotic plant cell, a prominent organelle is the chloroplast, the photosynthesizing particle that converts light energy into origin of life 157. 3,500 million years ago. (158 EVOLUTION) Evolutionary biology. Chemical energy. The following 2 hypotheses have been proposed regarding the origin of semi-autonomous organelles of eukaryotes, i.e., mitochondria and chloroplasts. A. Endosymbiotic hypothesis. One of the most intriguing speculations is that mitochondria and chloroplasts arose not via a gradual evolutionary process but abruptly in an unusually striking manner, i.e., symbiosis. In fact, mitochondria have a number of interesting properties that advocate that they were once free-living or independent, bacteria-like, i.e., prokaryotic organisms. Mitochondria have small amounts of their own DNA. This DNA exists as a loop-shaped molecule like the DNA of bacteria. Mitochondria also possess the genetic capacity to incorporate amino acids into proteins. These properties have led, in 1970, Lynn Margulis of Boston University to propose serial symbiotic hypothesis or endosymbiotic hypothesis. He hypothesized that mitochondria may have been derived from primitive aerobic bacteria, i.e., Paracoccus, that were engulfed by predatory organisms, probably fermentative bacteria, destined to become eukaryotic, figure 12.8. The predatory hosts became dependent on their enslaved mitochondria and the latter, in turn, became dependent off their hosts. Thus, the association was of mutual advantage to the predatory cell and the engulfed prey. Such a close association or partnership is called symbiosis. In essence, the mitochondria, formerly oxygen-respiring bacteria, established permanent residence within hosts. These predatory hosts became the first eukaryotic cells, i.e., animal cells. Chloroplast AWI. Eukaryotic plant cell. I equals O mitochondrion. Mitochondriona _ blue-greeny. Algae on _ J. B. Eukaryotic animal cell nucleus. Pipe. Aerobic. Bacterium % prokaryotic cell, anaerobic bacterium. Figure 12.8. Evolution of eukaryotic cell through symbiosis. The eukaryotic animal cell may have arisen through an endosymbiotic relationship between a fermentative prokaryotic cell and an aerobic bacterium. The eukaryotic plant cell may have originated by a comparable endosymbiotic relationship between a blue-green alga and a eukaryotic animal cell, after Volpe, 1985. Eukaryotic plant cells contain both mitochondria and chloroplasts. Just as aerobic bacterial cells can be equated with mitochondria, blue-green algae cells are basically equivalent to the chlorophyll-containing chloroplasts. Chloroplasts, like the mitochondria, have their own unique DNA and the associated protein synthesizing machinery. In this case too, it is speculated that those predatory cells, i.e., fermentative bacteria, that engulfed but did not digest the photosynthetic blue-green algal cells and the aerobic bacteria evolved into eukaryotic plant cells, figure 12.8. Thus, the engulfed prey became permanent symbiotic residents, either as mitochondria or chloroplasts, within the predatory cell. B. Invagination of surface membrane hypothesis. Many authors are not convinced with the hypothesis suggesting the relatively abrupt origin of the organelles of eukaryotes by a process of origin of life 159. Symbiosis. According to them, the origin of mitochondria and the evolution of living organ plasma membrane isms normally proceeds through a series of gradual changes, thus, continual modification of the surface membrane plasma membrane may have been the basic evolutionary mechanism, B. Origin of eukaryotic cell. In the differentiation of eukaryotic cell from prokaryotic cell, the organelles of eukaryotic cells may have evolved by the invagination, or drawing inward, of the surface membrane of a primitive cell. Figure 12.9 explains the postulated mechanism for the origin of a mitochondrion. From the cell surface memfig. 12.9. Hypothetical scheme of the origin of eukaryotic organelles from the surface membrane. A. Development of the mitochondrion from the surface membrane. B. Origin of a eukaryotic cell by the invagination of the surface membrane in several places, after Volpe, 1985. Mitochondrion. 243 Mitochondrion. DNA nucleus chloroplast. Brin. In support of this scheme, it is interesting that the enzymes for the breakdown of carbohydrates for energy production in bacteria are incorporated in the structure of the cell surface membrane. In similar manner, other specialized internal structures of eukaryotes, including the membrane-enclosed nucleus, may have differentiated from invaginated cell surface, figure 12.9. Finally, one of the new twists contributed by modern molecular biology is the possibility that today's prokaryotes are very highly evolved cells whose evolution has made them the most flexible and efficient cells on the planet. MOIECUIAR evolution. Evolution is a process of change. At the molecular level, this process involves the insertion, deletion, or substitution of nucleotides in the DNA. If the DNA encodes a polypeptide, these events may cause a change in the amino acid sequence. Over time, such changes can accumulate, leading to a molecule that bears little resemblance to its ancestor. Comparisons of the amino acid compositions of proteins in present-day organisms enable us to assume the molecular events that occurred in the past. The more distant in the past that ancestral stock diverged into two present-day species, the more changes will be evident in the amino acid sequences of the proteins of the two contemporary species. Viewed another way, the number of amino acid modifications in the lines of descent can be used as a measure of time since the divergence of the two species from a common ancestor. In principle, protein molecules incorporate a record of their evolutionary history that can be just as informative as the fossil record. Thus, proteins can be considered as chemical fingerprints of evolutionary history, because they bear amino acid sequences that have changed as a result of genetic changes. The evolution of proteins. A few thousand kinds of proteins are required for the functioning of an E. coli cell, and about 1 million kinds are required by human beings. They offer a preferable field for evolutionary study than 160 evolution evolutionary biology. Other macromolecules such as nucleic acids, because proteins are more heterogeneous both structurally and functionally, and are easy to isolate for the analysis. Recently, a number of proteins have been characterized by the method of sequence analysis. The common procedure for establishing the amino acid sequence of a polypeptide requires, first, breaking the protein into small fragments of peptides, and then determining the amino acid sequence in each peptide. The polypeptide is broken into fragments with enzymes, e.g., trypsin and chymotrypsin, that hydrolyze the bonds between contiguous amino acids at specific sites. The resulting peptides are separated from each other using such procedures as column chromatography and two-dimensional paper chromatography. The amino acid sequence in each peptide is ascertained usually with the help of an apparatus known as a protein sequencer, or sequenator, CFJ Ayala, 1976. Sequence data are now available from diverse biological groups ranging from microorganisms to mammals. Comparison of sequences allows study of interrelationship between structure and function, and to deduce how proteins have evolved. The laws governing evolution of proteins are similar to those for heritable traits, and interrelatedness of different organisms points toward their descent from a common ancestor. In general, the following features have been noted. 1. Identical proteins are not found among different living species, but homologous proteins with some similarities occur in diverse organisms. 2. Different positions in the amino acid sequence vary with respect to the number of amino acid substitutions that can take place without impairing the function of the polypeptide chain. Thus, some positions allow more substitutions than others. 3. Due to the tightly packed three-dimensional structure of the protein, a change undergone by an amino acid situated in the interior of the chain is likely to affect residues in neighboring positions. Such evolutionary changes, therefore, occur in pairs. 4. Enzymes performing similar functions have similar structures. Examples of protein evolution. 1. Insulin. The first protein to be sequenced was insulin. Insulin is a protein hormone produced by the beta cells of the islets of Langerhans of pancreas, which participates in carbohydrate and fat metabolism, which composed of only 51 amino acids. Sanger and Thompson, 1953. Comparison of the amino acid sequence of insulin in a variety of mammals, including cattle, pigs, horses, sperm whales and sheep, shows that with the exception of a stretch of 3 amino acids, the protein is identical in each of these species. 2. Hemoglobin. In comparison between hemoglobin of vertebrates, E. Zuckerkandl 1965 calculated an average of 22 differences between human hemoglobin chains, O and B, and the similar hemoglobin of horse, pig, cattle and rabbit. It was, therefore, concluded that human hemoglobin diverged from those of animals by about 1 amino acid change per 7 million years. The rate of change of amino acids is, however, different for different proteins. 3. Cytochrome circa. Cytochrome cis, an ancient, evolutionarily conservative molecule that serves as an essential enzyme in respiration. It is relatively small protein of a chain length slightly over 100 amino acid residues, which apparently exists in all eukaryotic organisms. This ubiquitous protein has been extracted, purified and analyzed in more than 80 eukaryotic species. Fitch and Margoliouth, 1967, Dickerson, 1971. Cytochrome C has 104 amino acids in vertebrates and a few more in certain species lower in the phylogenetic tree. Since cytochrome C performs similar vital functions in all organisms, fairly rigid evolutionary limitations have been placed on the acceptance of chance alterations, or mutations, of the primary amino acid sequence, i.e., an evolutionary conservatism is maintained. Some amino acid residues of cytochrome C have not varied at all throughout time. In particular, origin of life 161. The same amino acids have been found in 20 positions, P chain. In all organisms tested, B chain. From molds to humans. Obviously, the 20 amino acids at specific positions are essential, and any substitutions at these sites are likely to interfere with the function of the enzyme. In fact, there is one region of cytochrome C formed by a sequence of 11 amino acids, positions 70 through 80, that has remained intact in all organisms. In Table 12:1, the O chain number of amino acid L. Replacements in cytochrome C of several species have been compared. Dickerson, 1971. Firstly, it is evident that the chromosomes of closely related vertebrates differ in only a few residues, or not at all. For example, there is no difference at all in the composition of the amino acid residues between humans and the chimpanzee. Secondly, the greater the phylogenetic differences, the greater the likelihood that the cytochrome composition differ. This becomes clear when one compares a vertebrate with an insect, i.e., tuna and moth, or an insect with yeast. Heme group. Iron. Structure of hemoglobin molecule. Neutral theory of protein evolution. Evolution of proteins have also been studied in certain other proteins such as myoglobin, O-globin, histone, carbonic anhydrases, fibrinopeptides, etc. Analyses of the amino acid sequences of polypeptides suggest that some evolutionary changes may be random. This fact led to the formulation of a theory by Kimura, 1968, called neutrality theory of protein evolution. This controversial theory assumes that for any gene, a large proportion of all possible mutants are harmful to their carriers. These mutants are eliminated or kept at very low frequencies by natural selection. A large fraction of mutations, however, are assumed to be adaptively equivalent, i.e., the rate of evolution is equal to the rate of mutation. Table 12:1. Evolution of cytochrome C. Source. Volpe, 1985. A. Although the neutral theory does admit a role for natural selection as a purifying agent, that is, as a force that eliminates harmful mutations, it has little or no place for selection in the positive Darwinian sense. In the neutral theory, species do not get better by fixing beneficial mutations, they simply do not get worse by fixing only neutral or nearly neutral mutations. For this reason, this theory is also called non-Darwinian theory of evolution. See Gardner et al., 1991. RUAYRY 0N01 8 DN